So, welcome. It's just past the top of the hour, and I can see that we've just hit 90 people having joined us at this seminar. So I think it's a good time to kick off. So welcome. I'm Rich Scott. I'm the Chief Medical Officer at Genomics England, and I'm also in the interim being the Chief Scientist while we wait for our new Chief Scientist, Professor Matt Brown, to join us, who will be joining in November. So I'm hosting today's seminar, and we're very lucky to be joined by Sunayna and Melanie, who are going to be talking. I'm just going to start by giving you a little bit of some sort of housekeeping rules, and just remind you about some of the sort of upcoming things, and tell you about how to ask questions and so on. So first of all, you will probably have been notified as you join, but we're recording this seminar so that other people can enjoy it later. In terms, and we'll publish that on our YouTube channel, so you'll be able to see it. The seminar's split into two halves, so we'll have each of our speakers and then about 10 minutes of Q&A. So Sunayna's going to speak first about congenital malformations related to ciliopathies, and then Melanie is going to talk about renal tract malformations. And after each one, we'll have a little section of Q&A, and then we'll round up at the end. If you want to ask a question, can you use the Q&A function that you'll see in your Zoom window? You can also, if you want, raise your hand in the conventional sort of electronic way, and we'll come to your questions as we go through. And as I say, we've got about 10 minutes at the end of each speaker session. The seminars, as those of you who have come before, are all given by people who've been working on the 100,000 genomes data set and the growing National Genomics Research Library data set, to share some of the findings that they've got. We have a mixture of academics and industry users. We've got a particular focus at the moment on in this group of seminars on findings of particular patient impact, and we're particularly interested in participant involvement in programs. So particularly if you are interested in nominating yourself or someone in one of your teams to give one of the upcoming seminars, we'd be really interested if there's a particular participant angle, either as I say, in terms of involvement or impact. We're really keen to hear from you, and you can see on screen how to get in touch with the screen with the team. In terms of upcoming events and the next few seminars, many of you might have noticed that tomorrow we at Genomics England got what we're calling an innovation showcase running throughout the day. You can see that there are 5 sessions. We'd be really excited if people could join any or all of those. You can see talking about some of the particular areas of focus for us at the moment: COVID, looking at the work we're doing with the NHS on whole genome sequencing in the genomic medicine service, newborn sequencing, our work in cancer, and our work on diversity, ensuring genomics is delivered equally well for all. So if you are interested in joining, we'll put a link in the chat where you can register and sign up. And then in terms of the upcoming seminars, the next seminar, which is on the 26th of October, we've got Thanos from the Genomics England team talking about some of the work that the team here at Genomics England and the genomic team in Edinburgh particularly have been doing on COVID-19. And we've also got John Bridgewater joining us to talk about fusions in 100,000 Genomes Project cancer participants. So do join that one if you can, and we'll give updates via Twitter. And then we've got a series of further seminars planned in the future, and you can keep up to date by following our Twitter feed. You can also keep in touch with us by reading our newsletter. You can log on to our research portal, and the links for these will be popped in the chat. You can watch in time this video, but also videos of previous seminars, and to hear similar talks. And then the final thing that some of you might have heard is a podcast that we're doing. Chris Wigley, our CEO, mainly hosts that podcast, and it's a series with really engaging speakers. So that is sort of a plug for what's ahead. Apologies, I can see in the chat some people are struggling with my audio. I'm going to get my audio out of the way so you can hear from Sunayna in just a minute. So Sunayna is a clinical genetics registrar in Leeds. She's studied at UCL and Imperial in the past, and she's currently undertaking a PhD in Leeds, and is supervised by Colin Johnson, Chris Inglehern, and Carmel Toombs. And you're going to talk to us, Sunayna, about ciliopathies and congenital malformations. So look forward to your talk, and do pop your questions in the Q&A slot.
Brilliant. Thank you so much, Richard. Can everybody see my slides okay?
I can, yes.
Perfect. So yeah, thank you very much for inviting me to speak in this research seminar today. So as part of my PhD, I've been working on trying to improve molecular diagnoses for patients with ciliopathies that were recruited to the 100,000 Genomes Project. Just working out how to move on, sorry. So that was my overall aim, was to see if we could develop strategies to improve diagnosis rates for the patients with ciliopathies that went into 100K. So in my talk today, I'm hoping to present the 100,000 Genomes Project primary ciliopathy cohort, and then the diagnostic workflow that we developed in my PhD. I'm going to present the molecular findings and diagnoses we managed to identify in this project, and then some lessons learned and value added to participants. So a brief background on ciliopathies. These are a group of rare disorders due to inherited defects of an organelle called cilia. So cilia are found in two different forms. There are primary cilia, which has been the focus of my PhD, which are found on the surface of almost every cell type and have diverse roles in homeostasis and cell signaling. And there are motile cilia, which have roles in mucociliary clearance, particularly in the lungs and nasal passages. On the top right, you can see an image of some retinal pigment epithelial cells, which I've stained for nuclei in blue and cilia in red. And you can see these are like antenna-like structures that project off cell surfaces. There are over 35 different ciliopathy syndromes, which have significant genetic and clinical overlap caused by mutations in more than 180 genes. And most of these are recessive conditions. They form a quite wide clinical spectrum, from embryonic lethal rare conditions like Meckel-Grieber syndrome, right through to more common single system disorders, including polycystic kidney disease and retinitis pigmentosa, which affect more like 1 in 400 to 1 in 500 individuals. And on the right, you can see the spectrum of common features in ciliopathies, which include retinal dystrophy, brain malformations, developmental delay, skeletal abnormalities, kidney problems, and lots else. So participants could be recruited to the 100,000 Genome Projects under different ciliopathy categories. And my focus has been on those recruited with suspected congenital malformations caused by ciliopathies. And that included 83 probands. They fell into 3 different domains. So the first was rare multisystem ciliopathy disorders, of which I had 24 individuals. Schubert syndrome, which is a neurodevelopmental ciliopathy featuring brain malformations, developmental delay, and associated features such as retinal dystrophy and kidney problems. And Bardet-Biedl syndrome, which is an obesity ciliopathy. They could also be recruited under the respiratory category with the motile ciliopathy, primary ciliary dyskinesia, or non-CF bronchiectasis, but this has been the subject of a different study done by my colleagues in Southampton predominantly. So altogether, patients with suspected ciliopathies accounted for around 1% of the rare disease arm of 100K, although we suspect there are lots of hidden ciliopathy patients potentially in other recruitment categories. So I know this is a very well-educated audience, about 100K, but I thought I would provide a very quick overview of how patients moved through this project to then explain our rationale for our research protocol. So patients were identified by NHS clinicians and underwent a consent procedure and were recruited to the study either in the rare disease arm or in the cancer arm of the project. Once they'd been recruited, their clinician entered HPO terms to reflect their clinical features. They underwent whole genome sequencing, and then that data had panels applied based on the HPO terms entered by their clinicians. So PanelApp is a crowdsourcing tool that provides curated gene panels for different disorders. Variants then were tiered, so assigned a likelihood of potentially being damaging against genes in the selected PanelApp panels, and I'm going to show you what those tiers were in a second. And then a list of tiered variants went out to diagnostic labs, and they were contracted to assess tier 1 and 2 variants in the first place. And once that assessment had been completed, they performed a completion document called a GMC exit questionnaire. So the GMC is the Genomic Medicine Center, which coordinated this process. And this contained any variants that they thought potentially could be causative of the condition the patient was recruited for. So here was the GEL tiering system. So 3 tiers of variants were provided. Tier 1 are rare protein damaging variants in genes on the panels applied. So these are the most likely to be pathogenic, so things like stop gains and frame shifts. Tier 2 were rare protein altering variants, so predominantly missenses in genes on selected panels. And Tier 3 contained any other rare variants in genes not on selected panels. But what's important to note was, especially early in the project, structural variants weren't routinely tiered, and single heterozygous variants in recessive genes weren't routinely tiered either. And ciliopathies are mostly recessive conditions, so this became especially important. So we developed this diagnostic workflow, starting with filtering of single nucleotide variants, annotation and analysis. And then when we found individual heterozygous variants in recessive genes, we came up with a strategy to look for alternative second hits to complete the biallelic mode of inheritance. We performed ACMG classification of variants and assessed the match for phenotype in order to be able to assign a diagnostic confidence category. So here's the first stage of our workflow, which involved filtering, annotation, and analysis of variants. So we began by extracting all variants tiered by Genomics England, as well as variants regardless of their tiering status, using custom Python scripts. So we began by applying PanelApp panels, and then candidate genes lists were also provided from local and international collaborators, as well as curated kind of ciliopathy candidate lists called the Cecilia Gold list. So initially we used the ciliopathy panel, and then further panels were applied according to entered HPO terms, such as the retinal disorders panel and the DDG2P panel, which is a compilation of developmental disorder genes that was put together by the DDD project. We used the ensemble variant effect predictor to annotate our variants, and then undertook some structured analysis. And I did all of this blinded to the GMC exit questionnaires, as we wanted it to be an independent analysis. So I looked at all the variants called tier 1 and 2 by the GEL pipeline, which is the same as what the diagnostic labs would be looking at. I looked at tier 3 variants and genes that I thought potentially could be related to the phenotype. And then I looked at rare untiered single nucleotide variants that fell into the following categories. So those that have previously been called pathogenic by ClinVar, those called high impact by variant effector predictor. So these are things like stop gains, frame shifts, canonical splice defects, and rare missense variants predicted to be pathogenic by multiple in silico prediction tools. Once we'd found single hits through the SNV pipeline, we came up with this strategy to look for different second hits for heterozygous variants in recessive genes. So this involved looking for structural variants on IGV, the integrative genomics viewer, and looking for potential novel splice variants. So here you can see, I'm going to show you some other structural variants we identified, but this was a patient who had a heterozygous pathogenic variant in BBS1 who sounded clinically like Bardet-Biedl syndrome, where we were able to find the second hit as a heterozygous deletion involving 2 exons of BBS1. And then we used an in silico prediction tool called Splice AI using a further custom Python script to extract variants predicted to cause novel splice defects, initially on the ciliopathy panel and then on the DDG2P panel. And we used the cutoff score of 0.5 as recommended by the authors to provide a higher probability of a novel splice defect. We then applied the ACMG criteria to classify variants into the 5 classes of pathogenicity and extracted HPO terms from the user interface lab key to see if we could assess the match for phenotype. We also checked parental segregation, where we had trios or parents also recruited to the project, and used the medical literature and OMIM to assess for fit for phenotype. And this was how we assigned our diagnostic confidence. So this depended on whether we thought there was a good match to phenotype, what the ACMG classes of the variants were, and whether the gene was known to be OMIM morbid or whether it was a candidate. So for recessive genes, we needed 2 pathogenic or likely pathogenic variants in a gene known to be OMIM morbid to call it a confident diagnosis, 1 pathogenic or likely pathogenic plus a VUS to call it probable, or 2 VUSs to just call it possible. And for dominant and X-linked genes, we needed a pathogenic or likely pathogenic variant for a confident diagnosis and 1 VUS for possible. Where we thought there wasn't a very good match to phenotype or the variant wasn't a candidate gene, we required 2 pathogenic or likely pathogenic variants in a recessive gene for a possible diagnosis and 1 in a dominant or an X-linked gene. So we didn't want to give any higher confidence than possible for that group. So prior to doing any genetic analysis, we looked at just the clinical features for patients entered to these 3 suspected ciliopathy groups to decide whether we thought they were likely to actually have ciliopathies as they were recruited. And for about 40% of patients, we thought that was probably unlikely. So we were looking for the presence of classical ciliopathy features, such as retinal dystrophy, polydactyly, cerebellar abnormalities, to give us a flavor of whether they were likely to have these conditions. And we did a bit of reflecting on why potentially they were less likely to, and wondered about the following. So this is slightly unanswerable, but we'd be curious about your thoughts as well. So we wonder if these conditions aren't terribly well recognized universally in the clinic, so whether they're over or under diagnosed. For some, we wondered if they'd gone into slightly inappropriate 100K categories. So we found quite a few patients that went into the Bardet-Biedl category who turned out to just have isolated eye disorders. So maybe they'd have been better going into an eye-specific category. And we think there's been some incorrect or incomplete HPO term entry, quite a lot of missing detail, which makes it much more difficult to interpret the variants that you find. So here's a summary of our molecular diagnosis rates. So in the pie chart on the left, I'm presenting the outcome of GMC exit questionnaires in the data release that I used in this project. So that's from data release 11. So at that time, the diagnosis rate from 100K was 32.5%, of which 23 cases were reported as solved and 4 with VUSs, but nearly 20% were unreported. So the cases hadn't been closed by the diagnostic labs, and there wasn't a complete GMC exit questionnaire. And by the time I completed my analysis, we had a diagnosis rate of nearly 52%, of which 28 we classified as confident, 5 as as probable and 10 as only possible. So we've achieved a nearly 20% diagnosis uplift through doing this project. We've also identified one false positive. So this was a patient reported to have a ALMS1 deletion, which we couldn't identify either on IgG or in BCF files. And on further dialogue with the 100K team, did conclude this was a false positive, which was fed back to the recruiting clinician. So then thinking about what kind of diagnoses we found, as suspected, we didn't find a very high rate of ciliopathy gene mutations. So nearly half the patients actually had non-ciliopathy disorders. So that was kind of surprising given they were recruited, suspected to have these conditions. And I'm going to show you a bit more of what we found. So here's a breakdown of diagnoses by recruitment category. So here you can see that for all three of our groups, we found variants in ciliopathy genes and in non-ciliopathy genes. The best diagnosis rate was amongst those in the Joubert syndrome category. And this is because we think that's got the kind of hardest handle phenotype. So Joubert syndrome patients classically have a brainstem and cerebellar malformation called the Muller tooth sign, which is pathognomonic for Joubert. So the pickup rate in that group was the highest and worst in the rare multisystem ciliopathy disorders category. This was kind of a catch-all term and only found 3 ciliopathy cases in that group. So an interesting spread of diagnoses identified. So here's some of the non-ciliopathy diagnoses found. So as I said before, we found 4 patients in the BBS category who have variants in genes that cause eye disorders but aren't known to be directly linked to cilia. So the question here is, have we only made a partial diagnosis and the clinicians really thought they had Bardet-Biedl syndrome but didn't enter those HPO terms, or are these just inappropriately in that category? And then here's some of the multisystemic conditions we identified, some of which we were quite surprised by. So, for example, in a patient recruited with suspected Joubert syndrome, we found a known pathogenic CHD7 variant in keeping with charge. And a patient with BBS, we found a RAI1 variant in keeping with Smith-Magenis syndrome. So not necessarily the most obvious differentials, but a really interesting spread of conditions. Then looking at the kind of variants we identified, we thought that nearly 40% of the diagnoses could have just been made from tier 1 and 2 variants, which is what's being looked at routinely in diagnostic labs. But for 11 patients, they had at least one causative variant outside of tiers 1 and 2, so would have potentially been missed. So that included 5 tier 3 variants and 12 that were untiered, of which 3 are structural variants and 9 are SNVs. And there was also 2 variants miscalled by GEL, so the false positive ALMS1 deletion and an indel miscalled as a tier 3 missense. So here's some of the structural variants we found. So the first one is that miscalled indel, which was called as a tier 3 missense. You can see it's actually a more complex indel taking out the beginning of an ALMS1 exon. And then we found 3 heterozygous deletions, 2 of which were multi-exon, and 1 was taking out a single exon of CSPP1. That's the case on the right-hand side. So in the proband, we actually only found the deletion in, I think, 7 reads, but it was present more like 50-50 in their father. So we collaborated with a diagnostic lab for this patient, and this deletion was confirmed experimentally in the lab and has been fed back as a likely pathogenic change. The patient also has a splice variant. Then looking at our Splice AI analysis, our custom Splice AI script revealed 3 variants, non-canonical splice, that would cause non-canonical splice defects. The first is a synonymous change in RL6, which wasn't listed on PubMed, but when we went to the medical literature, did find that it's been previously published in association with BBS, with functional evidence through a mini gene assay showing an aberrant splicing effect. And then we found 2 other variants just outside of the canonical splice sites that were predicted to be pathogenic also. We have identified a putative new ciliopathy disease gene. So this is a gene called LRRC45, which is on the Ciscilia Gold version 2 list, which is currently under review as a potential ciliopathy gene, but so far no patients have been found. So this encodes a protein which contributes to early steps of axoneme extension during ciliogenesis. And we found a patient from the rare multisystem ciliopathy disorders category who sounded clinically most like BBS with retinal dystrophy, developmental delay, and some structural brain changes, who had a homozygous frame shift variant in LRRC45, which was absent from NOMAD. And they have a sibling in the intellectual disability category who's also homozygous mutant, and both the parents are hets. We then used a script called gene variant workflow to extract variants in this gene across the whole of 100K and found a potential second patient. So this is a patient with what sounds like non-syndromic cone rod dystrophy, who's got a start loss and a splice acceptor variant, but we haven't been able to establish phase because we don't have any parental sequence. So final thoughts are about how we've added value to participants through doing this project. So as most of you know, having a molecular diagnosis allows us to provide much more accurate genetic counseling. And I think this is especially relevant as we've revealed so many diagnoses that we didn't expect to find. So most of these patients, if they were counseled that they were suspected to have ciliopathies, would have been told that these are recessive conditions with the most likely 1 in 4 chance of having further affected children. And if we can show that they actually have a different condition with a different mode of inheritance, that really changes how we counsel them. Having a molecular diagnosis allows us to offer cascade familial testing, prenatal testing, and PGD where appropriate, and allows us to direct patients to appropriate support groups. Also allows us to offer any available screening or treatment if that exists, and provides a foundation for development of targeted therapeutics. One patient I thought was especially interesting was participant 61, who was recruited to the rare multisystem ciliopathy disorders category and had an unsolved GMC exit questionnaire. And this patient had predominantly structural renal problems. And through our Python scripts, we found an untiered missense variant that's previously been listed on ClinVar as pathogenic, a gene called WT1, which is associated with structural kidney problems and an increased risk of Wilms tumor. So we managed to feed this back to the recruiting clinician who alerted the patient to the diagnosis, and they're now receiving screening for early detection and treatment of Wilms tumor should this develop. So I think here especially we've proven how important it is to get the right answer for these patients. So in summary, the diagnosis rate here from whole genome sequencing is nearly 52%, of which just over 13% might have been missed by only looking at tier 1 and 2 variants. And this shows the value of collaborating with researchers who have a bit more time and facility on hand to go digging in this amazing data set. Nearly half the patients didn't have the conditions we expected according to their recruitment categories, which encourages us to consider wider differentials and open up the search to other panels where that's appropriate and data is available. And I've got lots more 100K analyses ongoing. So I've done some extensive ciliopathy gene, candidate ciliopathy gene analysis across the data set. And I'm currently preparing a manuscript for a reverse phenotyping study where I've been able to identify about 20 hidden ciliopathy patients in other categories through that gene variant workflow script. So this data has recently been accepted for publication in the Journal of Medical Genetics. We don't yet have a publication date or DOI, but we will disseminate that when we have it. And if you want to use any of our scripts, they are freely available through GitHub, have mostly been written by Jenny Lord in Southampton. So I'd like to thank my supervisors and my funding body, the Wellcome Trust. Thanks to Genomics England and lots of people at the help desk there who I'm constantly pestering with queries, and my colleagues in Leeds, particularly from Team MECL and the Vision Research Group. And I'm happy to take any questions.
Thank you, Sunayna. That's brilliant. So just a reminder to people, the best place to put questions is in the Q&A thing, which is the little thing with 2 speech bubbles at the you can also put them in chat. And I can just see we've got a question from Evgenia, which is on partly sort of looking particularly at that WT1 case as an example, Sunayna, but I guess the sort of a general theme about saying how much have you been able to go back to look at the phenotype and sort of understand more about some of these surprising findings, and what are the lessons from that?
So I found the main barrier actually is not having very much clinical data, and I found collaborating with recruiting clinicians more difficult than I'd hoped. So for all these new findings, I've sent out research identified diagnosis forms through the airlock, but I've only had a kind of handful of responses, which is a bit of a shame. So I don't know if that's a systems problem or a lack of response problem. But if anybody has had those queries from me, I'd be really happy to hear from you. I think the more clinical data we've got, the better we can interpret variants, which I'm learning a lot now. But I'm doing this reverse phenotyping study that it's hard to properly interpret variants without proper phenotyping data there. But I haven't heard back from everybody yet, unfortunately.
Yeah, and on that, so we at Genomics England have and are increasing the support for that, so that the sort of speediness of that Recontact process. And we're also in discussion with NHS England about how best to support that, because we know, for example, that some clinicians have moved on and so on. So it's about, or are just very busy. And it's about how we work out the best support for that, because we want to make sure that that interface is as easy as possible to navigate.
Yeah, I've been talking to Helen Britton at GEL, who said that when this paper is published, it's going to go out to all the recruiting clinicians with a notification saying number 62 is yours. And hopefully that means that all these results get properly disseminated.
That's brilliant. We've got a question from Omid, who is asking, have you compared your findings to Xmiser hits? So Xmiser is an algorithm that we now use, didn't throughout the 100,000 genomes, but in later cases, later participants, and in sort of our ongoing pipeline to help prioritize based on HPO terms and so forth, and variant frequency, etc.
Yeah, I looked at Examizer right at the beginning, but because it's not fully comprehensive, we decided to go down the line of extracting all variants and then annotating them ourselves and filtering them ourselves, because I was worried if I used Examizer, I would just miss things. But it would be interesting to go back and compare to see how good the matchup was.
And that's really useful to do. And one of the things that I know some of the other groups have done, and in dialogue with NHS England, is look at what are now the reporting guidance that NHS labs have when they're looking at whole genome sequence, which have evolved on the basis of work like yours and others. So, for example, where at the beginning of the 100,000 genomes, there was a more focused look, now people are, in general, looking for de novo variants, less relevant to ciliopathies, but relevant to some of the participants that you've been looking at, and looking at top 5 Examizer hits and sort of working out where that sweet spot is in terms of making as many diagnoses as possible.
Yeah.
So Ahmed has asked, did you include genomes aligned to GRCH, so genome build 37 as well?
Yes. Yeah, I did both.
Yeah. And just for context, probably the first fifth or a little bit less of cases in the project went through on that build 37.
Yeah, definitely.
Then we've got a question from Michael. Did you find many cases with single heterozygous changes that look like a clear phenotype, but unable to find the second variant, with just for sort of people less familiar with ciliopathies, with the commonest pattern being an autosomal recessive, so where you'd expect to find a pair of variants in the same gene on the maternal and paternal copies?
Yeah, I have a very frustrating list of what sounds like very convincing phenotypes with single hits, and I just can't find the other change. So there's got to be other things we can look at, whether it's a more comprehensive look at structural variants or intronic variants predicted to affect splicing. But yes, I have got a list of single hits left to go. I'm working now with a colleague in Oxford called Jing Yu, who's developed a structural variant extraction script called SV-Rare. So he has a database of rare structural variants called by Mantan Canvas and 100K. So we're looking for the ones that are only called very rarely to see if they could give us an extra clue. But so far I've solved one further Bardet-Biedl case with a homozygous deletion. So there's still lots, obviously, we can extract here. It's just knowing how to do it.
Great. And then I wonder whether we can get Patrick to ask his question. Patrick, you put a question in the chat. I don't know whether Sam and team, you can see if Patrick is able to unmute to ask his question.
Hello. I'm Patrick. Hi. So I was just really interested in how you managed to take untiered variants in your pathway and kind of return them into tiering. So getting variants which weren't in tier 1, tier 2, or tier 3. Did you have any particular lessons from this? Just it seems like you managed to make diagnoses, and I think it was about 9 or 12 patients where they didn't have tiered variants. And I was just interested in that you just use ClinVar pathogenic variants as well, because I know that ClinVar can be quite an unclean database, and variants which are clearly pathogenic can sometimes be marked as having sort of conflicting evidence statuses. Thanks. Hopefully that makes sense.
Yeah, so when we were looking at untiered variants, I think the majority of them came up because either they were single hits in recessive genes, or they were in genes not on the panels applied so far. So that's how we found most of them. And then ClinVar, you're right, isn't a very clean system, but it's just a way to develop a pipeline that you can apply to lots of cases. But it's absolutely not comprehensive, and the conflicting views mean that when we found anything, I did try to go properly through the medical literature to see if there was better evidence out there beyond just a ClinVar listing, but it was just a way to try and pull out anything potentially new and exciting.
Thank you. Valentina asks, can you expand on how you systematically screen for structural variants? Only recessive genes, or did you, where there was a first hit, or were there other things that targeted where you were looking for them?
So I think that now, looking back on it, having worked on this data set for the last year and a half, this is the least scientific bit of this strategy, which is basically when I found a single hit, I pulled up the region on IGV and manually trawled through to see if I could just spot anything. As I said, I'm now developing, Jing in Oxford is developing this new system to do it in a more systematic way. There is structural variant calls in 100K from Mantan Canvas, but my problem is I've never really known how to filter them because you get so many calls, of which when you look at them manually, most of them look like rubbish. I don't have a clear system like I would for single nucleotide variants to filter down to things that could be pathogenic. So I'm aware this is a not very sensible way of doing it, but I was completely new to whole genomes analysis when I started this at the beginning of COVID. So I'm sure there are much better ways of doing it than how I managed.
Yeah, and that, for context, that remains hard. It's one of the reasons that in 100,000 genomes we didn't include those in tiering, because of the amount of false positives, just as you've seen, sort of overwhelming numbers. But there have been quite a lot of successes through work like yours and, for example, the work on the pilot project, looking in a more targeted way to help you to find areas where there were likely to be hits. And now in our active clinical pipelines, we go down to a 10 kb size for variants, but still, it's still a sort of work in progress. It's still hard, isn't it? And then linked to that, Santosh says for the second hit on these biallelic genes, we've got one variant of suspicious nature. Were you looking at regulatory regions like promoters, enhancers, or?
I haven't, and that might well be another source of second hits. So yes, if anybody has any clever ways to teach me how to do that, I'd love to. But we needed to draw a line and try and get this published. So no, I think that would be a really interesting source of potential further diagnoses.
Right. And then I guess one question that occurred to me as you were talking, one of the big themes here is that these ciliopathy disorders are hard to spot both ways, as in you saw that people recruited participants as under the ciliopathy category where that wasn't the case. And likewise, you did some work and can see that there are a number of participants with clear ciliopathies recruited under other categories. One of the things that NHS England have done through their test directory is actually move away from a sort of a phenotype-led testing strategy to feed the relevant participants into sort of broader ranges of genes being looked at, so not just ciliopathies but broader. Are there any other lessons that occur to you that could sort of that could be helpful for guiding clinical care?
I think the more comprehensive phenotyping we can get, the better. So I've got 4 cases that went in as Bardet-Biedl listed with only retinitis pigmentosa, where I have eye disease gene mutations, but I will never know whether they are a partial diagnosis because the clinician really thought they had BBS, or that was the whole story. So the more comprehensive phenotyping you can get, the better. I've just had an interesting case come up through my next project, the reverse phenotyping strategy. So this is a patient that went into the epilepsy plus other features category, where I found compound heterozygous CEP290 variants. Patient just was entered with epilepsy and keratoconus. And when I went back to the recruiting clinician, turns out they've also got labor congenital amaurosis and cerebellar abnormalities. So that completely changes how I interpret the variants. So really, the more comprehensive phenotyping we can get, the better for interpretation. Oh, you think you're on mute, Richard? Sorry.
Thank you. I said that is a great lesson to end on. Thank you very much. I think we've answered the questions that have come up in the Q&A, and this has been a really engaging talk and great Q&A session as well. So thank you very much for joining us. Now we're moving on to our second speaker, who is Melanie Chan. So Dr. Melanie Chan is a clinical research fellow at the UCL Department of Renal Medicine and a registrar in nephrology. And she's currently in her final year of her PhD, using statistical genetic techniques to analyse whole genome data from the 100,000 Genomes Project to understand structural kidney and urinary tract malformations. So Melanie, thank you for joining us, and look forward to hearing your talk.
Great, thank you very much, Richard, and thanks for asking me to come and speak about some of the work that I've been doing as part of my PhD. So as Richard mentioned, I'm a clinical research fellow, so I'm very much a clinician, and currently undertaking my adult nephrology training in North London. So I'm going to start by giving an overview of posterior urethral valves, or PUV, and explain why this condition is one of the biggest unmet clinical needs in paediatric nephrology. I'm then going to discuss some of the different types of population-based association analyses that we've been carrying out, and show how these experiments have given us some novel insights into the genetic architecture of this rare disease. So PUV, or posterior urethral valves, are the most common cause of kidney failure in children and young people. They only affect boys and occur in 1 in 4,000 male births. Now, the valves are these obstructing membranous folds that are found down here in the posterior urethra and cause congenital bladder outflow obstruction, leading to a buildup of urine in the bladder, the ureters, and the kidneys, which can cause damage. Now, treatment involves ablation or removal of these valves in infancy, but long-term complications can arise because of associated kidney dysplasia, where the kidneys don't form properly, as well as bladder dysfunction, where those affected have to self-catheterize in order to pass urine. Now, up to 60% of those affected will develop kidney failure before the age of 30, which means to survive, they often need to spend many hours a week on dialysis, which has many associated risks, or they need to have a kidney transplant. And this places an enormous burden both on the affected child and their family. Now, at present, we don't know what causes PUV or understand what goes wrong during development of the urinary tract whilst in the womb. Although it's usually sporadic, clustering within families and some twin studies have suggested that there might be a genetic component to PUV, but as yet, no robust genetic associations have been identified. So to try and better understand the genetic basis of this rare disease, we use the rich data set generated by the 100,000 Genomes Project to perform some population-based association testing. Now, one of the key advantages of using this data set, which sequences obviously your cases and controls on the same platform, is that you can produce a really nice clean signal because you're minimizing the risk of confounding by technical or sequencing artifacts. So as a very brief reminder, population-based association testing means we take a group of cases, a group of controls, we sequence both of those cohorts, and essentially are just comparing the DNA between the two groups, looking for differences in the genome. We can look both at common variants, so those generally seen in more than 1% of the population, or rare variants, which we can actually collapse down into groups or genes before comparing between our cases and controls in what are called collapsing tests. Now, these differences in the DNA or in the genome can give us insights into the underlying biology of disease and potentially lead to the development of targeted therapies. So in our study, we used whole genome sequencing data from 132 male unrelated cases, none of whom had received a monogenic diagnosis via the project, and just over 23,000 ancestry matched controls who were made up of the unaffected relatives of non-renal rare disease participants who had no clinical evidence of kidney disease or kidney failure. Now, our cohort, the median age was 14 years, and you can see that only a small proportion, 4%, had a family history. We went on to analyse both the burden of rare single nucleotide and structural variation, as well as common and low frequency variation, but using single variant association testing, or GWAS, as well as an independent replication study. So given the small number of available cases with PUV, we made the decision to actually include all ancestries in our analysis in order to increase our power to detect association. Now, this is a slightly unusual approach, given that a major concern in genetic association studies is that population structure can lead to false positive associations caused by differences in allele frequencies between the different groups. I think as a field, we are now increasingly aware of the European bias in genetic association studies, where, for example, we know that almost 90% of all GWASs are carried out in individuals from European backgrounds. So with this at the back of our minds, we also wanted to explore whether we could adequately control for population structure in a mixed ancestry group, using some of the newer statistical approaches that have been recently developed. So to account for this population stratification in our mixed ancestry cohort, we used two approaches. First, we matched our cases to our controls using the top 10 principal components, using a weighting algorithm written by Adam Levine, who is a renal histopathologist in our group. Now, this principal components analysis, or PCA plot here, just shows the differences between the first two principal components, but illustrates the variance seen between all of our samples corresponding to their different ancestral backgrounds. Here we have the blue matched cases with the black matched controls, where each case had to match at least two controls to be included, whereas we discarded two cases here that you can see in orange and all of the grey controls. The second approach we used was a tool called SAGE, which is available in the research environment. And this models how genetically similar each pair of individuals are and incorporates that information in a logistic mixed model to account for both population structure and sample relatedness. An additional benefit of using this approach is that it can adjust for case control imbalance, such as that seen in our cohort, which can also lead to false positive associations. So, numerous attempts have been made to identify candidate genes for PUV, but as yet, no robust associations have been identified. So, to try and identify some novel candidate genes in an unbiased way, we use these collapsing gene-based approach, where we collapsed rare, likely deleterious variants together by gene across the entire exome and compared our cases to our controls. Now, this is a Manhattan plot showing the chromosomes along the bottom, the strength of association along the y-axis, and each of these dots represents a gene. Dots below the line are those that you would expect to see purely by chance. Now, you can see that we did not identify any single gene to be enriched for rare variation in our cases exome-wide, suggesting that there was no evidence for a strong monogenic component to PUV. So given we hadn't detected any rare variant associations or monogenic associations, we next wanted to investigate the contribution of common and low frequency variants to PUV. And to do this, we performed a genome-wide association study of 17 million variants with a minor allele frequency of greater than 0.1%. Again, here you can see the Manhattan plot, but rather than these dots representing genes, these now each represent a single variant. The red line here represents conventional genome-wide significance of 5 × 10⁻⁸. So this time, we can see that there are 2 regions in the genome that differ between cases and controls, one here on chromosome 6 and one here on chromosome 12. Now, the QQ plot and the lambda of 1.05 indicate that the population stratification was well controlled in this mixed ancestry group. We also carried out conditional analysis to try and identify any secondary independent signals, but didn't find any. And there was no evidence of any epistasis or interaction between the lead variants at each of these loci. So having identified these signals, we needed to make sure that they are true. And to do that, you carry out a replication study. So we did this in an independent European cohort consisting of almost 400 PUV cases from Germany, Poland, and the UK, as well as 10,800 controls who were taken from the cancer cohort of the 100,000 Genomes Project, excluding any of those who had a urological, renal tract, or childhood malignancy. And none of these individuals had been included in the discovery analyses. In our replication study, we genotyped the lead variants at both of the genome-wide significant loci, as well as 2 further loci that had suggestive evidence of association. We managed to confirm association of both of the genome-wide significant loci, although with smaller effect sizes than had been seen in the discovery cohort, which we think likely reflects the smaller sample size of our discovery cohort, as well as a mixture of winner's curse and the differences in risk in the non-European populations. The 2 suggestive loci did not replicate despite being adequately powered. Now, looking at the variants in a little bit more detail, the lead variant on chromosome 12 is a common variant with an allele frequency in controls of 37%, and this has a protective effect, whereas the variant on chromosome 6 is a rare variant with an allele frequency of just under 1% with quite a large effect. And it should be noted that this signal would have been missed if we had used a conventional array and imputation panel, and demonstrates the utility of using whole genome sequencing for genome-wide association studies. So having replicated these 2 loci, we just then wanted to check to see whether we would have been able to find them if we had just looked purely at Europeans. So we repeated our GWAS just using genetically defined Europeans for 88 cases and just under 18,000 controls. Here you can see that the chromosome 12 locus just remained genome-wide significant, but the lead variant at this locus showed weaker association than that of the mixed ancestry GWAS. The chromosome 6 locus here was not detected this time, demonstrating that by including individuals from different ancestral backgrounds to increase our sample size, it can be an effective way to boost power even in a small cohort. We next wanted to kind of dive a little bit deeper and try and identify the likely causal variant or variants at each of these loci, which is a process called fine mapping. Now, the great thing about whole genome sequencing is obviously it lets us look in detail at these regions, which again is not possible with your conventional arrays and imputation. So whereas previously we had looked at low frequency and common variation, we then repeated the association testing at both of these loci, including all of the variants, to determine whether there were any additional rare variants that we were missing. However, both of the lead variants that we detected previously remained the most strongly associated, confirming that the signal that we had observed included the likely causal variant. We next applied a statistical fine mapping tool called Painter, which integrates strength of association, linkage disequilibrium patterns, as well as different functional annotations to show that both of these lead variants that we've detected had a very high posterior probability of being causal. So through validating both of these lead variants using statistical fine mapping, we've shown that whole genome sequencing has great sensitivity and resolution and may actually allow us to directly identify the likely causal variant. This is especially relevant when we're looking at rare variants and non-European populations, when imputation can be less accurate. Now, we know that the different populations display different patterns of linkage disequilibrium, or LD, which is when alleles are more likely to be inherited together than by chance. Now, these different patterns of LD can be very useful to narrow down association signals and are commonly used in transethnic meta-analyses. This figure here illustrates data from the 1000 Genomes Project for European, South Asian, and African populations at both of the genome-wide significant loci. The black triangles indicate haploblocks, or groups of alleles that are normally inherited together, whereas the yellow arrows indicate our lead variant. You can see here that the pattern of triangles or haploblocks is different in the different population groups, showing that by including these different ancestries, we can take advantage of this naturally occurring differences in LD to help pinpoint or narrow down likely causal variants. So having fine mapped these regions and identified the likely causal variants, the next key questions are, what are these variants doing? Which genes are they affecting? And what are their functional relevance? Focusing first on the most significant variant in chromosome 12, you can see here this is a locus zoom plot with our lead variant seen as a purple diamond. Just remember that this is a common variant with a protective effect. Now, this variant falls in an intergenic region, so it's a non-coding variant, but was shown to intersect with a highly conserved region and actually had a really high, pretty high CAD score for a non-coding variant of 15, suggesting that it is likely to be deleterious. Using publicly available chromatin interaction data from embryonic mesendoderm cells, which are the precursor to the bladder and the proximal urethra, we then mapped this region to the promoter of the nearby gene TBX5, which is a transcription factor. Now, heterozygous variants in TBX5 cause something called Holt-Oram syndrome, which is characterized by both upper limb anomalies and cardiac defects, demonstrating that this gene is already known to have a role in embryonic development. Interestingly, the risk allele of this lead variant has been associated with prostate cancer, which, given PUV is a defect of the developing prostatic urethra, makes an interesting link between the developmental and cancer pathways. This is the latest release of FinGen GWAS, and you can see here that although it's not significant in this one, there is suggestive association of this variant with, again, prostate malignancy. And there is a significant association with female genital prolapse, which was replicated in the UK Biobank, where you can see that this variant is associated with both urinary incontinence and genital prolapse. So this provides additional support for an association with this variant with lower urinary tract phenotypes. Now, the likely causal variant at the chromosome 6 locus is found in an intron of this gene PTK7. And this is a rare variant with an odds ratio of 7.2, so a large effect size. Now, this variant was found to intersect with a transcriptionally active region in mesendoderm cells, as well as transcription factor binding sites, suggesting it may have a regulatory role. PTK7 is known to be a planar cell polarity gene, which regulates embryonic development via the non-canonical Wnt pathway and has been linked to neural tube defects and scoliosis before. So with a collaborator of ours at Manchester University, Professor Adrian Wolff and his postdoc, Philippa Lopez, we then went on to carry out immunohistochemistry in a 7-week gestation human embryo to look for expression of these two proteins that we identified via the GWAS. So at this stage of development, the mesonephric duct here is seen here, and this will form the base of the bladder, as well as insert into the prostatic urethra to form the male ejaculatory ducts. The urogenital sinus seen here is the precursor to the embryonic bladder and the proximal urethra. This shows PTK7 staining. As you can see, the brown staining is quite intense in the stromal cells surrounding the mesonephric duct, also seen in epithelial cells surrounding the urogenital sinus. And the TBX5 was detected in a nuclear pattern in a subset of epithelial cells around the urogenital sinus. So this demonstrates that both of these proteins can be detected in a normal developing human urinary tract. So up till now, we've been focusing on single nucleotide variants and small indels, but we know that structural variants, so defined as those larger than 50 base pairs, affect more of the genome than any other class of variation. And they've been associated with a wide range of diseases, including congenital anomalies. Now, historically, SV detection has been limited by the available technology, but we thought we would capitalize on what was offered by whole genome sequencing, such as better resolution and sensitivity, as well as the opportunity to investigate balance rearrangements, such as inversions, which would be missed by conventional microarrays. So in our analysis, we pulled out rare autosomal structural variants, so deletions, duplications, inversions, as well as copy number variants that were both gene disrupting, so those that intersected with an exon, as well as those that intersected with cis regulatory elements. We compared the overall SV burden between our cases and controls, as well as on a per gene or per regulatory element type basis. However, using this workflow, we did not identify any enrichment of gene disrupting structural variation, either overall or affecting any single gene, suggesting that this is probably not a common mechanism in PEV. We did, however, see an enrichment of rare structural variants affecting cis regulatory elements. Now, this is a slightly busy graph here, but it shows the type of structural variant along the top, the type of cis regulatory element along the bottom, and the proportion of individuals who had at least one structural variant along the y-axis. Now, we identified a significant enrichment of duplications affecting proximal enhancer-like elements, as well as of inversions affecting many of the different types of cis-regulatory elements, but in particular, CTCF-only elements was most significant. And this was not something that we had seen when I had looked at the rest of the CACA cohort, which comprises over 800 probands. Now, CTCF is a chromatin loop anchor, and one of the best explanations we could come up with to explain these findings is that potentially these inversions are disrupting chromatin looping or chromatin conformation, and this disrupts the long-range regulatory networks that are necessary for urinary tract development. So these are quite preliminary results, but they raise some interesting questions, and we'd like to do some further work to validate these associations and better understand the exact role of structural variation here. And obviously, long-read sequencing, I think, would be really, really useful here, so we could better delineate these inversions and try and validate this signal. So in summary, we have shown that whole genome sequencing is a really useful tool to investigate the genetic architecture of rare disease beyond conventional monogenic gene discovery. We have identified both common and rare variant associations with PEV and demonstrated that common variation can also contribute to an individual's risk of rare disease. Specifically, the rare variant in PTK7 would have been missed if we had used standard genotyping and imputation. The increased burden of structural variants affecting regulatory elements is also interesting, but requires a bit more investigation. we've demonstrated that you can use a diverse ancestry approach, which is commonly seen as a problem in genetic association studies. And we can actually use that to increase power to detect new disease associations, even in a small cohort, and leverage the differences in the LD patterns to enhance fine mapping and identify likely causal variants. And finally, we have identified 2 novel susceptibility genes for PUV, TBX5 and PTK7, which are the first robust genetic associations of this disorder and will provide insights into the underlying biology of this disease. I'd like to thank my supervisor, Professor Daniel Gale, as well as Professor Bockenhauer, Adam Levine, and Omid Siddiqui Alavijay from my group for their help with this work, as well as our collaborators in Manchester and the University of Bonn, the Genomics England Bioinformatics team for all their support, the patients and families obviously involved in the project, and my funder, Kidney Research UK, who has funded this research. If you're interested in reading anything more about this, then our preprint is now up on medRxives, and you can scan the QR code to access that. But I'd be happy to take any questions. Thank you.
Thank you, Melanie. Fantastic talk, really interesting, and sort of a real, as you say, really interesting what you can do beyond sort of rare conventional rare variant analysis and looking at burdens and so on. We've got a couple of questions already in the Q&A function, so do keep them coming in the background. So first one from David Curtis, which was the gene that fell just short of genome-wide significance that you mentioned. He's interested to know whether it's biologically plausible.
In the rare variant association.
I expect so.
Yes, yeah. So the gene, I think, is TMRU, which is a mitochondrial transfer RNA. And my very brief looking into it was that no, it did not strike me as biologically plausible.
Great. And David, do say if you've got if you'd like to come in with any further questions. Sam and Zan, I wonder if unmuting David just in case he's got any follow up. Oh, that's good. He's Q&A'd, so that's fine. And then Ines is interested to know which data sets you used to carry out the functional fine mapping with Paintal.
So we used I used cis regulatory elements from ENCODE. I think I used DNase hypersensitivity from the embryonic mesendoderm cells. So some of them were specific to mesendoderm cells and some of them were generic. So I think like the transcription factor binding sites that I used were generic, but that was a...
Thank you. And just, you're still sharing your screen, Melanie, so if you just come off... Am
I? Okay.
That's great. And then Shamima was just coming in, just wanted to check whether that was TRMU that you mentioned. And I don't know whether Shamima, you want to just talk a little bit, as it obviously a gene close to your heart.
All right, it's interesting.
Hi, I don't have a lot to add there, but just to say, Melanie, great talk, really enjoyed it. And there are quite a few mitochondrial disorders with renal manifestations and a few that have been linked to CACUT. So I don't know if they'd be of interest to you.
Yeah, I'll definitely have a look at it. As I said, I had a kind of a brief literature search of the gene because obviously it was my top gene, but nothing jumped out at me. But I will have. Do you know specifically what type of CACUT? This is asking specific question.
Oh gosh, I must have been one person. I can find some papers and send them to you. Your email was... That would be really great. UCL, so I'll find you. Yeah.
Brilliant. Thank you so much.
Okay.
And I guess Melanie, a sort of follow up from me slightly on that point. Like Shamima, as not a renal physician, when I see people in clinic who have some sort of urinary tract malformation, I guess sort of led by some of the data on HNF1 beta, etc., I sort of tend to think actually one structural renal anomaly sort of might be associated with another with the same gene. I guess in my mind, posterior urethral valves maybe do like they're filed in a different section? Is that like that based on sort of, is there, you know, what's your sense of the truth here? Is posterior urethral valves quite different, or you talked about looking at this in the context of the broader CACUT group of participants, 100,000 genomes? Does that make sense? Would you find the same hits, etc.?
Yeah, I mean, that's a really interesting question. So as you say, I've been looking at the kind of entire CACUT cohort, and you're right, it's a very heterogeneous cohort, both genetically and phenotypically. So the same gene can cause a multitude of different phenotypes, you know, as you see with HNF1 beta. Posterior urethral valves I mean, there's no monogenic causes that have been identified so far. You saw in our cohort, I think only 4% of them had a family history. So it's not classic. It seems to be more of heading towards more of a complex disease, a polygenic risk, rather than obviously a monogenic rare disease. Although interestingly, it is quite often associated with kidney anomalies. So there must be some shared developmental pathways that are going wrong. Because I think in our cohort, there was about 20% of them had associated renal dysplasia or cystic dysplasia. And there was actually one patient who had an HNF1 beta, a 17q12 deletion, which didn't quite fit with the phenotype. But when we went back to the clinician, it turned out that they did have all the features of HNF1 beta disease. So they had cystic renal dysplasia, they had, I think they had hypomagnesemia. So we wanted to double check the phenotypes, and it kind of appeared like they almost had two separate phenotypes, because I'm not sure that the HNF1 beta has been associated with, well, it hasn't been associated with PUV before. So we think that was a case of two distinct diagnoses in that patient.
Right, and then I think we've got one hand up I've been alerted to. Is that, I don't know whether Zan and Sam can unmute.
Absolutely, yeah. We take what we need. Exactly.
And is it Jean or Jean who, I don't know whether you can be unmuted.
Good.
Don't worry, if not.
Good. I mean, always feel free to ping me if you need to.
We've got one more question in the Q&A, and otherwise we can come back to that person. So Prasen asked a question about the math you use for your structural variant part of your study, whether that was a data set that you developed or calculated from the 100,000 genomes data, or whether there was some external source of that data.
No, so we calculated that based on the entire case control cohort that we used. I didn't mention this, but what we actually did is we also used the cancer cohort because that was about, I think, 10,000 genomes to generate a set of common variants, so common structural variants. And we also used the NOMAD common structural variants. And we filtered on that. So we excluded all common structural variants that were found in the cancer cohort or were found in NOMAD. And then within our group, we then calculated the allele frequency based on the cases and controls together. Because actually within the 100,000 genomes project, I think we've got, NOMAD is based on 15,000 genomes. So, we've got more genomes that we can use to calculate allele frequencies.
Thank you. And then for your reading offline, Melanie, I can see that Shamima is like peppering the Q&A.
I saw that. Great, thank you, Shamima.
Follow-up data. And Ruth Newbury just has put a comment in about Holt-Oram syndrome, just sort of highlighting the lack of renal malformations in Holt-Oram patients from her experience.
Yeah, and I guess obviously Holt-Oram syndrome is associated with rare missense or protein truncating variants. So whereas the variant that we found that was associated is a common variant, and it's protective. And I thought it was, very interesting that the same variant is associated with both prostate cancer, which again is the same kind of, we're looking at the prostatic urethra and prostate cancer, and also, urinary and genital phenotypes in adults. So we also know that it's expressed in the right place. So I think it's just a different mechanism of disease, essentially.
Yeah, thank you. So just checking in again whether there's anyone else with any questions before we thank Melanie for her talk. That has been really, really interesting, and I think kind of quite nice and complementary to the sort of very monogenic start from Sunayna, and then a more complex genetic picture, both of which sort of with sort of really interesting angles in terms of how you can use genomes to understand what's going on. So thank you again, Melanie, for a really fantastic talk, and all the best with the remainder of your PhD.
Thank you very much.
So thank you all for joining us today for those two excellent presentations. Just as a reminder that this is a regular series, so our next sort of talks on the 26th of October are Thanos talking about COVID-19 and John Bridgewater talking about fusions in cancer participants in the 100,000 Genomes program. So please do join us for the next last Tuesday of the month for this seminar series. And then also just a reminder that please sort of stay in touch with what's up in the sort of research community and genomics England more broadly. The links Sam has just posted again in the chat. And we really enjoy the dialogue, both through these sorts of seminars and through the various other channels. So really enjoy that. And I think some of the themes that come out through these seminars are sort of themes that we're really aware of more broadly across our work. For example, Melanie, they're talking about the challenges of making sure that findings from studies like hers are relevant to everybody, not just people of European ancestry. Sort of a mixture of sort of ensuring that we are aware of the lack of diversity historically in genomic data sets, but also the need to develop new tools and sort of know-how in terms of making sure findings are relevant. So thank you again all for joining us, and looking forward to seeing you next month at this series, or if not before, in one of our other events. Thanks again for joining us, and a really big thank you to Melanie and Sunayna for two really, really interesting talks.